we've had a lot of really a lot of interest in the course and a lot of input. Gabe showed the GOC. I guess it's a community of practice of individuals that have been interested in this, and nothing is an original idea. So it's been great to see that we've had this idea to try and structure this program from everybody on the working group there, and we're having a good time planning this, and we look forward to your feedback to make improvements, and we're having fun doing this. So I have to say that I don't have any conflicts to declare. I am not going to talk about synthetic lethality or BRCA mutations. They've been covered elsewhere, and much better than I can cover it. And so I'm kind of focusing this part of my talk on the questions that have just been asked about mutation significance, because I mean, it's something I struggle with on a daily basis to try and put this into perspective and how we use this genomic information to help patients. And so I'm going to try and just provide a bit of clarity, but also to say that like I'm far from having any brilliant answers for you. When it comes to this slide, I think somebody wondered, why would I put up this slide? A bunch of the mutant Ninja Turtles, and the title of my talk relates to this, but in actual fact, the Ninja Turtles, they're ninjas in feudal Japan, were responsible for espionage and sabotage, and they were actually discredited by the more honorable fighters in Japan at that time because they waged irregular warfare and used underhanded tactics to fight. And so in a way they're very similar to cancers. And I think you can only postulate what sort of mutations have led to this phenotype, but I think it's there's a heavy dose of because I'm interested in proteins, there's a heavy dose of obviously hormone receptor-mediated cofactors here that's interacting with the mutations to cause this phenotype. So I think one of the things that I want to talk a little bit about, though, is this relationship to cancer phenotype, which has become so complicated by the number of mutations. So we're trying to struggle with the significance of a particular mutation in a cancer with a very complex phenotype. And in the case of BRCA1 and 2, we started off with the phenotype. We know these families developed cancer, but we're kind of coming at it from the other way around when we describe these mutations, and we're trying to figure out what they mean. One thing that we heard that was really interesting this morning from Dr. Huntsman was just this, because when I was a medical student, this was anything causes cancer, it's this complex dynamic between DNA damage and DNA repair, and it's mediated by age and inflammation and what you eat and other carcinogens and this sort of thing. So, but we heard this morning that there are now signatures of chromosomal changes and genomic signatures that actually relate to the etiology of the cancer, whether it be inflammation or microsatellite instability, as a result of DNA mismatch repair genes and that sort of thing. So this is really interesting and might help us further subclassify cancers and get a little bit more at like what causes these things. Because as a clinician, I'm often asking myself, well, why does ovarian cancer have 96% of them have a P53 mutation? Like, how did that arise? So these are the interesting things, and I'm not going to be so bold as Trevor was to put up my own karyotype, because then I'm definitely abnormal. So, but Trevor already went over the more traditional and the new ways of classifying this. So I'm going to go right into talking a little bit about oncogenes, and I think this is in clinical, for those of us that do clinical care, this is one area where the confusion has reigned, because this is a fascinating story that resulted in a Nobel Prize discovery, and like our initiatives to develop this course, there were actually 3 groups working at this at the same time, and there was a merging of different strategies for studying cancers, because the first one being that they knew that viruses cause cancer, and a group had already been able to determine that there were certain genes within retroviruses that were responsible for transforming cells, in the laboratory. And then at the same time, groups were using already bona fide cancers, and they were from bladder cancers. The group was studying how bladder cancer DNA could be transfected into the same types of cells and resulted in the same kinds of transformations in these cells. And then lo and behold, they put the two together. The one group decided that they would actually use a plasmid that had incorporated the viral DNA as a probe, and they probed these bladder cancer cells that had been transfected with other human DNA, and lo and behold, they found both a gene that was responsible because the NIH3T3 cells were mouse cells, and but they were transfecting it with human DNA, and they found two different size fragments after digesting, this DNA and probing it with, they had a radioactive plasmid probe that they Nick translated, and they were able to see that there were two fragments, and that led them to believe that one was the mouse gene and one was the human gene, and that the virus had actually incorporated human genes that were similar to these genes that cause cancer in that the viruses resulted in. And then subsequently, in contrast to the viral gene that just strictly overexpressed this oncoprotein in the human, they found that there was just a single point mutation that resulted in the transformation. So it was really an amazing discovery, and the other group that was working on it, ended up getting a Nobel Prize, and similarly the same investigator, I think it was Weinberg, he also was the first to describe the retinoblastoma tumor suppressor gene, I think. So we got into this concept then of oncogenes actually causing cancers, and we got into thinking that mutations are the key driver in cancers, and it's true. Either a mutation in a gene that's an oncogene, and these are usually point mutations that result in the gene being activated, constitutively activated, usually in proteins that mediate growth and differentiation, like key regulatory signaling proteins, and then tumor suppressor genes that we're familiar with, and certainly, the BRCA1 and 2 story are the big story that's been a success on all levels. But we know when these genes are then mutated that normally if they're regulated properly, there's a normal cell, but if they become mutated or overexpressed, or there's a translocation that results in the overexpression of that gene, then this can lead to cancer. So the classic theory that still holds true in the majority of situations is still this progressive accumulation of mutations, but more recently it's been appreciated that there can be catastrophic rearrangements in the genome that result in cancer that aren't sequential, but they may occur just as a single catastrophic rearrangement, so-called chromothripsis. It's not felt to be as common as this, but these changes are sequential, and we need several of them, and this has been proved in mouse models. And recently, for example, there's been a paper just showing that, if you have a single ARID1A mutation in a mouse model, that doesn't lead to cancer, but you need additional mutations to cause cancers in mice. So this continues to be a theory that's recognized and proven over and over again. Now, when we look at these oncogenes that are responsible for causing or being associated with the development of cancer, and so the reason I talk about this is because it's very different from us thinking about how we're going to target or treat a cancer, but these are responsible for developing a cancer. There's now, you can see the growing list of genes that are now associated. It's gone up to more than 500 that may confer a survival advantage to cells and dysregulate them to the extent that they can develop cancer. And Trevor already touched on this, so I'm not going to go into this in detail, but there's a lot of heterogeneity, and the good thing for us as gynecologic oncologists is that actually the common types of mutations, we can actually keep track of them, because the mutations that occur with any frequency, and for us that oftentimes oftentimes people joke that, because we're surgeons, we can only keep track of 6 or 7 pieces of information at any one time. And so this is good for us because we can kind of keep track of these common mutations that occur and distinguish, different cancers by virtue of their mutational profiles. And so this has become something that is mainstream with the different histotypes. And so for classification purposes, this has become very important. But if you notice, there's all these other thousands of mutations that occur as Trevor's gone over, and like how do we put those into perspective as potential therapeutic targets? It's a huge challenge. And when you look at just ovarian cancer, specifically, we have the key mutations that are described, the P53 mutations, BRCA1 and 2, and then there's 5 or 6 others that occur with any degree of frequency, 6%, 3%, and the rest of them are, this huge genomically unstable tumor that presents these treatment challenges and challenges in terms of finding targets for treatment. So, but the area where it's really helped us is in classifying, but also in understanding the precursors of various types of cancers that arise in the gynecologic or in the genital urinary tract. And so as an example of this just shows you the different P53 mutations. And so this is just the codon distribution of the mutations and the types of mutations that occur, and then the mutation patterns in terms of, exchange of base pairs and whatnot. And I mean, there can't be anything really more complicated than the types of P53 mutations that one can describe in cancers. And that's all cancers that's reflected in that slide. Oh, I think I'm missing a slide here. No, it comes next. So, but when it comes to studying these things, and Trevor's far more of an expert on this, and he alluded to the importance of bioinformatics, but then it becomes, if we identify a specific oncogene or a specific mutation in cancers, like how do we know that it's really relevant in terms of not just the start of the cancer, but potentially as an oncotarget? And does this mutation actually influence the function of the protein? And so there's now like a huge field of in silico or computer modeling where these mutations can be modeled. They can be compared against the various SNP databases to rule out the mutation being a functionally relevant mutation. The changes in the sequence can be compared across different species to determine whether it's really a relevant mutation in terms of causing cancer. And then in silico modeling of protein structure as a means of predicting protein function. So like these statistical methods are very important, and they're used in the Nature papers that have tried to analyze sequence data in gynecologic cancers, the TCGA data, and trying to put some of these mutations into perspective. But when it comes down to it as well, it's really important, as biologists that are trying to understand the relevance of the mutation, that these have been described in the context of the various hallmarks of cancer that are dysregulated by mutations in cancer. And so, scientists have developed a number of different assays to try and describe whether these mutations are having an effect on the phenotype of the cancer, whether it be, evading growth suppression or dysregulation of their cellular energetics, evading immune destruction, the genome instability. So there's various assays to determine whether these mutations actually have functional significance as hallmarks of cancer. And the list of these hallmarks have expanded into stressors of cancer, things like the fact that the cell has to respond to a genomically unstable environment, and perhaps these can be used as targets for the cancer. You probably think this is odd. I'll ask one question at this point, is what kind of gynecologic cancer cell is this? What's that? A granulosa cell, yeah. And I have to apologize for the color, but this was done. The color is, with all respect to Prince, who passed away within the last couple of weeks, and who's probably one of the most underrated guitarists that ever lived. But I changed the color in his memory. So we have this paper then to lead on and follow up with the P53 mutation story, because interestingly, this has allowed us with such a heterogeneous nature of mutations. When they discovered that the same cancer, the same 53 mutation was in the fallopian tube as was in the actual cancer, I mean, it was significant. What's the probability that that would be the same on the basis of chance with the hundreds and hundreds of different P53 mutations that just naturally occur? So it has been helpful. Mutation analysis has definitely been helpful for us to describe precursors and understand how these cancers have developed, and understand that in this case as well, that cancers that have an ARID1A mutation actually arise from endometriosis. And so I'm just going to spend a few minutes on this story because we've done a little bit of work on this. But in reference to the frequency of mutations, when the ARID1A mutation was discovered by Dr. Huntsman's group and another group on the East Coast, the fact that it occurred in 45% of clear cell cancers and 35% or so of endometrioid ovarian cancers was like a home run. And because it was such a common mutation, and so people were really excited about this as being a potential target. But what it actually led to, it's a very complex protein. In fact, the function of the protein is regulated within the context of this SWI/SNF chromatin remodeling complex. And like there's over, what do they say, there's over 29 genes that are responsible for regulating this particular function, and it's not just one. So this is how complex it gets when we're trying to determine whether a specific mutation is actually having a functional impact on a cellular regulatory process of importance. So, and as it turns out, when we looked at when these mutations were looked at in the context of cancer, I mean, what was really probably the most intriguing thing was that these mutations have been described in associated endometriosis. And so the same mutation was found, because once again, there's a wide spectrum of mutations, this being a tumor suppressor gene, wide spectrum of mutations that occur within this particular gene. But the same mutation has been found in the adjacent atypical endometriosis that was next to the cancer. This is the ARID1A. This is just it's a depiction of the different genes that actually have been mutated, but ARID1A is at the very bottom here. And you can see there's slight expression on whole exome sequencing for ARID1A in the adjacent atypical endometriosis and in the adjacent endometriosis, suggesting that this tumor has in fact arisen from the endometriosis and the atypical endometriosis. We looked at this a little bit further because in the original New England Journal paper, there were only 2 cases that they described whether they noticed the ARID1A or they found ARID1A mutation in the associated endometriosis. And so we explored another 35 cases and reviewed the literature to see whether or not this was the case. And we just used the immunohistochemistry for BAF250, which is the protein that's expressed by the gene. And you can see here's just an example. And the nice thing about BAF250 is that we can see it in the normal. It's recognized within the nucleus of the normal tissues. And so we can use that as an internal control. And you can see here in the endometriosis, here's endometriosis, which is positive for BAF250 or does not have an ARID1A mutation. And here we have atypical endometriosis that's positive for BAF250, an example of atypical endometriosis that's negative. So we looked at a number of clear cell and endometrioid cancers, 35 of them, and examined each cancer for loss of BAF250 in the adjacent endometriosis. And this is a compilation of not only the work we did, but also from 2 other studies, because there's now been over 110 cases that have been described. And we found BAF250 loss in the cancer in about 55% of them. And in the endometriosis or adjacent endometriosis, a significant number of those had loss in the adjacent endometriosis. And on the flip side of it, the BAF250 was retained for the tumors that BAF250 was retained in, there was no loss, no cases of loss in the adjacent endometriosis. And there's no cases of loss that have either been described in isolated atypical endometriosis or endometriosis remote from the tumor. So it's just an example of how a mutation has allowed us to describe the development of this cancer and the fact that endometriosis, these tumors are arising from endometriosis. Perplexingly, however, though, what about all the ones that don't have an ARID1A mutation? So, and interestingly, when we compared proteomic analyses using reverse phase protein array, so we looked at about 100 different proteins that were involved in signaling and signal transduction and receptor tyrosine kinases. We found that when we compared the tumors that were ARID1A deficient or BAF250 deficient versus the ones that weren't, we didn't really see any differences in protein expression between the two tumors, suggesting that the mutation's not having a significant effect on the phenotype of those cancers. And you can't distinguish them when you look at them, histologically. They look exactly the same. And intriguingly, and somewhat unfortunately, there's no difference in prognosis when you compare ARID1A tumors with ARID1A mutant tumors with non-ARID1A mutant tumors or wild type. So it doesn't, unfortunately, it hasn't really proven to be any kind of a prognostic or predictive marker. So Trevor had mentioned this little list of tried to put together a little list of targeted therapies that have actually, are now being used in the clinic. And there's a couple of interesting examples here. And I want to spend a few minutes just talking about an area that we've been studying, and that's an area we've been looking at MEK inhibitors, and in conjunction with Gabe, we've been able to, in the OVCare program, we've been able to generate a number of cell lines from low-grade serous cancers that we've been able to study. But if you look at the successes that have been described to date relating to targeted therapies, well, here's a imatinib, and this is a fusion protein. You're familiar with that story. Trastuzumab, which is for breast cancer associated with a 3 to 5% overall survival rate, but this is a copy number variation that's resulted in successful treatment. Cetuximab in GI cancers, metastatic GI cancers, and it's used now in lung cancers, but 5-month median survival, but only effective in tumors that are wild type for KRAS, but this is an overexpression, not a mutation. VEGF, as we're all too familiar with, unfortunately, in ovarian cancer, which wasn't a very successful treatment. Again, not even any biomarker used, just treating ovarian cancer, but trying to target angiogenesis. And then this has been a success with melanomas, the dabrafenibs and the afatinibs that treat a specific mutation for BRAF in melanomas. And this is a proven success story where there was no previous treatment that had any benefit at all, a PFS of 5 to 7 months and a response rate of 50 to 60%, which in melanomas was unheard of prior to that. And then this is an interesting story recently because this only, for lung cancers, this ALK fusion, another fusion protein, it's only responsible for found in about 5% of lung cancers, but quite a significant response rate. So a success as a targeted therapy. And then I'm going to just spend a minute on this, a new drug, AZD9121, and I'm just going to talk about this one for a minute because it's a bit of an interesting story. Before I do that, I'll talk a little bit about the work we've done on the MAP kinase pathway because we've studied a number of MEK inhibitors, so this is just an outline of the pathway, and it gets rather complicated because, interestingly, RAS is not a kinase; it's actually a GTPase, and its function is rather complex in terms of how it's activated. It hasn't been a real success story in terms of developing inhibitors for KRAS because of this complexity, but these pathways are, often shown like this, but they're actually, far more complex in terms of the way that the signaling works. And as Dr. Huntsman had pointed out this morning, in many instances, it's cancer or cell type specific as to how important the signaling is for regulating the oncogenicity of that particular cell. I want to go back one. Did I get it? Yeah. So this is the BRAF story, and they had an opportunity to, use BRAF inhibitors in the MAP kinase pathway, and even in studying a small number of cell lines, you can see here that with the mutation, these are the IC50s, or the inhibitory concentrations that inhibit 50% growth of the particular cell line. Here we've got like just complete inhibition with the BRAF mutation, whereas the few tumors with NRAS were somewhat sensitive and somewhat not sensitive, and then the wild type definitely not sensitive. And when we look at the downstream effectors of the pathway, which is ERK phosphorylation, you can see that the drug actually inhibits ERK phosphorylation. And interestingly, the baseline phosphorylation status didn't really correlate with outcome at all. And nor was there any obvious correlation with how effectively the drug inhibited ERK phosphorylation, other than to say that it did. What's that? 2 minutes. Okay. So when we looked at our own cell lines, we've studied our own cell lines in this regard using MEK inhibitors, and so we've seen some similarities. Like first of all, you've heard the big debate yesterday, as far as the low-grade cancers were concerned, about whether RAS mutations are either predictive or prognostic. And Dr. Gershenson, outlined how complex and poorly understood the pathway was. We've actually seen within the context of these are the different cell lines we tested, we've actually seen that it's not just cell lines that are involved with mutations. Here we've got 2 cell lines that are KRAS and 1 that's NRAS, so it's a small number. But nonetheless, in all these cell lines, there's actually, quite remarkable changes in copy number that involve the MAP kinase pathway. So it's not just a mutation that might be mediating these effects. But we've obviously seen some differential effects based upon this individual cell line. Here's one that's completely sensitive to 4 different MEK inhibitors tested, and here's one that has differential sensitivity to the different MEK inhibitors tested. And again, the baseline level of ERK phosphorylation didn't correlate. But we did find there was definitely differences in drug. And this is an interesting example that I just show you, because this particular drug, Dr. Gershenson mentioned yesterday that the study that was using this drug for treatment just closed. It didn't reach its midpoint evaluation for effectiveness. And this drug actually has never really been tested in low-grade cancers, low-grade serous cancers. But if you look at, this is dosing at 24 and 72 hours. But if you look at the degree of ERK phosphorylation in the resistant cell line here, you can see that it doesn't inhibit phosphorylation to the same degree that trametinib does. And so, like, we were able to at least see in cell lines that perhaps, one of these drugs is better than another, which begs the question that perhaps these drugs should be, more well tested in cell line models before they're taken into the clinic. I'm just going to finish with this now because I'm running out of time, but this, actually, I'm going to move on to the next example because I think how am I doing for time there, Gabe?
You're done.
I'm done, okay. So let me just go back to this here. Well, this is just an example of the ALK mutation, and I just want to make one little point in relation to this, because in terms of targeted therapies, the concept of a targeted therapy working is based upon this concept of oncogene addiction, whereas the ability of the malignant tumor or the cells to proliferate really is based upon the need for that cell to have this mutation as a driver. And so this is a very different concept than oncogenesis. Now we're talking about oncotherapy, and I would suggest that probably a more appropriate term, because even oncogene addiction isn't really an an easily understood concept, nor is it sometimes biologically relevant. But when we think about oncotargets, I think we need to develop some new paradigms in terms of how we study these to determine whether a mutation is really an oncotarget. And I think we're just at the start of doing this, and obviously a complexity is the tumor heterogeneity, because that really makes it a complicated thing. But in this particular case, I'm just showing you with crizotinib, I mean, what happens is it does work for about 6 months, but then these secondary mutations have been described in the tumor that displace the drug from the binding site, and then the tumor is no longer responsive. One thing that's fascinating, and I'll just finish with this, because I just touched on this whole idea between oncogenesis and prognosis, and I think we all are kind of left scratching our heads sometimes when we end up talking about pathologic classifications, and we're into reclassifying tumors that now have a different prognosis, but they have a variety of different treatments. And how do we figure out whether treatment, now that, and the example this morning were these POLE mutations in endometrial cancer, but we don't know how they're treated or what stage they are. So how do we make sense out of whether this is really going to allow us to judge new therapies or the elimination of treatment without further study? And I'll just finish off with an interesting example. I was talking to you a little bit about this new drug, the AZD9121, because it targets a secondary mutation that develops, an EGFR mutation that develops as a result of the EGFR, of the cetuximab treatment. So this isn't the same protein, but now they've developed a drug because for maybe the first time, and this drug is a winner because in lung cancer it has a 12-month, they still haven't reached the median survival, but they actually, to select the phenotype based upon a prior drug treatment would suggest that drug now, that is an oncotarget, because the tumors really had to mutate to escape the inhibitory effects of the treatment, and now they've come in with a tyrosine, or now they've come in with another EGFR inhibitor that actually can inhibit the secondary mutation in the tumor, which has resulted in probably one of the most lengthy tumor success responses and durations of remission that we've seen with a targeted mutation treatment. So I'm running out of time, so I'll finish off at that. And just to say, yeah, this not to discourage you, but I mean, because this question did come up, when we apply some of these, mutation profile screening and whatnot, like we're still at the beginning of trying to determine how to apply these, and it may only benefit a small number of patients when we screen for, do mutation profile screening in patients and try and use that to select treatments for them.